Thank you, Jeff. That's quite the introduction. Well, please don't call me an oceanographer. Keep me into trouble today.
You are one.
I spent time at sea, that's for sure. Anyway, thank you for inviting me. It's actually a really an honor and a pleasure. And I think this will be a lot of fun. My primary goal today is to tell you a little bit about what we do in our lab with the hope that this inspires some of you, the people that I don't know as well, to walk down the hall, knock on my door, and tell me that you're not doing this, but if you were, it could be even cooler. So come seek me out and have a discussion and go into some of this in more detail, and use this as an opportunity to learn a little bit about what our lab can do and how that may interface with your interests. So we're going to be talking about a lot of things today. I wasn't aware of the fact that there's going to be some spats of omic-related talks in this venue. That's what we do, so we're going to talk even more about that today. I'll try to make it gentle, if possible. We'll see about that. I'm still struggling with that myself. So we're going to talk a little bit about methods. That's useful. We're going to talk a lot about these things called oxygen microzones. Places in the ocean that don't have a lot of oxygen. It's one of the primary focuses of our lab. How microbes are shaped, how ecosystem processes are shaped by the presence or absence of oxygen. So we're going to spend some time thinking about what our meta-omic data tells us about that. Then we're going to also address this idea of niche partitioning. What are the environmental factors, microspatial factors to some extent, that determine where microbes are in the ocean and what they're doing in the ocean. We're going to look at this over vertical gradients, things that are easy to sample, the meter scale, the tens of meter scale. We're also going to address some data related to a much finer scale. Microspatial variation between bacteria that are free living in the water column versus those that are hanging out on organic particles or inorganic particles. What do those two different niches mean in terms of who's there and what they're doing? Okay. So this is a bit of a mixed crowd, and I thought I'd start with this slide that I always start with, just to remind people a little bit about the world that we live in. It's a microbial world, right? So this is data from what is now a bit of a classic paper by Barney Whitten, University of Georgia. In the late 90s, he tried to put total cell count on different major biomes out there in the oceans, terrestrial surfaces, the terrestrial subsurface, and he had tried to attach estimates of carbon content, nitrogen content to these bugs out there in the environment. Trying to get some sense of just how diverse are prokaryotes, or how ubiquitous and abundant they are. Prokaryotes, bacteria, and archaea, small cell, single cell. These are some of the numbers that he came up with, somewhere on the order of 10 to the 30 total cells, total prokaryotic cells, out there on our globe. These numbers have been changed a little bit. These were found not too long ago. But the basic picture stays the same. There's big repositories of microbial life, abundance, in places that we don't sample so often, the deep terrestrial subsurface, that's both beneath the continents and beneath the oceans. It's where you find a large, perhaps the largest fraction of microbial cells. Not necessarily the largest fraction of microbial activity in life, so we also tried to assign estimates of turnover time, bacterial doubling time, and some of these, of course, are speculative and based on some wild extrapolations. And so a lot of the activity isn't focused there in the subsurface, but there's a lot of activity in other places that I care about, especially in the marine environment, especially by photoautotrophic organisms, heterotrophic organisms, in the surface waters of the marine environment. We're going to talk a little bit about those waters and some mid-water depths. Like I said, you also attached some estimates of elemental composition, elemental pools to these numerical numbers. Came up with some of these values, and if you add up all of the microbial carbon out there, it equates to about 50% of what you find in terrestrial land. It's a big fraction. It's even bigger if you look at nitrogen and phosphorus. 10 times the amount of nitrogen and phosphorus is locked up in prokaryotic cells compared to terrestrial organisms. So I show this data just to set the stage, that we are living in a microbially dominated world, and to steal a line from Paul Kalkowski and Ed DeLong, these microbes are really the engines that drive the Earth's biogeochemistry. That's why a lot of people in this room care about them. That's why I care about them to a large extent. This is data from Adam Martini's group looking at the distribution of single-celled photoautotrophs for Chlorococcus, one of the most abundant prokaryotes, photosynthetic prokaryotes out there in nature. These bugs and a lot of other microbes are doing tremendously important things. They recycle key elements, analyze organic carbon. They do other things as well that we really care about. They oxygenate our waters, oxygenate our atmosphere. But it hasn't always been that way, right? This is data from a paper in 1996, looking at Earth's history, the distribution of oxygen content in shallow parts of the ocean and deep water parts of the ocean. For long periods of Earth's history, the ocean is effectively anoxic, right? There's still life there. This is during the time when most of the wide diversity of microbial functional types, protein types evolved. A lot of the metabolisms that microbes are so versatile at evolved during this period of anoxic oceans. And you can still find, to some extent, analogs of those ancient oceans today. To some extent, maybe some of the same microbial processes. And so we study these things called oxygen minimum zones. I'll get to a slide in a second that shows the diversity of different oxygen minimum zones. I'm going to focus on some big ones. Permanent oxygen minimum zones. There are about three of these, three big features, three oxygen minimum zone features in the world's oceans, one in the Arabian Sea, and then two in the eastern boundaries of the Pacific Ocean, off the coast of Chile and Peru, and off the coast of northern Mexico. These two sites are where we've done most of our work. These are big features, thousands of square miles. Mid-water depth features that hang out roughly over a depth profile of about 500 meters in the middle of the water column. Big water masses. Like I said, they come in all shapes and sizes, these oxygen minimum zones. This is a review paper that we published a couple of years ago, highlighting some of the diversity in oxygen minimum zone types out there in the oceans. If you go anywhere in the oceans, off the coast of Hawaii, very well oxygenated water column, you're always going to find some intermediate depth zone that has a minimum oxygen concentration. We can call this an oxygen minimum zone. It's not the type that we focus on. But here, oxygen goes from maybe 200, 250 micromolar at the surface to, I don't know, 50 to 100 micromolar at this zone of lowest oxygen concentration. That doesn't matter, really, in terms of microbial life, in terms of what microbes may turn themselves on or off in response to oxygen. 100 micromolar oxygen, 50 micromolar oxygen, tons of oxygen. Right? So these features aren't so interesting in terms of thinking about microbial responses to oxygen depletion. There's more drastic depletions of oxygen that occur in some seasonally anoxic parts of the ocean. This would be an example of, for example, the dead zone that you find in the Gulf of Mexico at the mouth of the Mississippi each year. Oxygen pretty high at the surface can drop quite low in the dead zone, for example, below 20 micromolar in some places, below 5 micromolar in other places. And oftentimes you see this 20 micromolar as kind of the defining boundary for where you start getting into a true OMZ. But still, 20 micromolar oxygen, that's plenty for a lot of microbes, for a lot of microbial processes. One of our goals is to try to figure out where along that gradient of oxygen depletion does it actually start becoming important. We start seeing shifts in microbial function. Is that very dependent on, of course, what the process is and the taxonomic group carrying out that process. So for answering those questions, we're working in zones like this. These are zones that are emblematic or characteristic of those three big OMCs that I showed you a couple of slides ago. Oxygen again at the surface, 200, 250 micromolar. It decreases steeply, and it decreases a lot. So at the core of these OMZs, oxygen anywhere from 15 to 50 nanomolar. Effectively anoxic. 3 orders of magnitude depletion in oxygen concentrations. This is a big shift, a big environmental gradient. And the magnitude of that shift is going to come into play a little bit later when we think about the key factors that are structuring the microbes and where they're hanging out and what they're doing. So these are the type of zones that we've been focusing on in the Eastern Tropical Pacific. We call these anoxic oxygen zones. We abbreviate as AMZ for short. And what happens along that oxygen gradient in these AMZs? This is, again, a image from this review paper that we published a couple of years ago, just highlighting some of the key functional processes that occur. A lot of stuff going on. A lot of things related to the nitrogen cycle. We'll talk about this in a second on the next slide. In both the reductive direction as you're going down in this graph, and also in the oxidative direction as you're going upwards. There's also processes that are coupled to the sulfur cycle, the sulfate reduction, and sulfur oxidation by very diverse groups of microbes in that zone. And all of these, well, a proportion of these metabolisms are heterotrophic, of course, mediated by bugs that are using organic carbon as an electron donor, but a lot of these processes are autotrophic as well. The reason for me mentioning it is that to emphasize that some of these nitrogen cycle and sulfur cycle processes are directly coupled, of course, to the carbon cycle, directly coupled to the sulfur cycle as well. A lot of OMZ research has focused on what happens in terms of nitrogen in OMZs. We've been thinking about this for a long time. They've been thinking about it in different ways, not necessarily applying a lot of meta-omics to it, but thinking about it in other ways, using either single gene approaches or process rate measurements to try to figure out what are the different processes that occur in these cells. And there's a lot. And why do we care about OMZs in particular in relation to the nitrogen cell? It's because they're anaerobic. Because they're anaerobic, certain types of nitrogen metabolism can take place. And two of the ones we care a lot about are these processes of anoxic, which is the anaerobic oxidation of ammonia to nitrite, a terminal electron acceptor, and this process of denitrification, oxidizing organic matter with oxidized nitrogen species, or nitrogen species. Both of these processes are very important in the OMZ zones, in particular because they result in the production of nitrogen gas, right? Not only nitrogen gas, but also N2 gas to some extent. And we care about these, right? These can bubble out of the oceans. As a consequence, these processes represent nitrogen sinks. And so we care about that. There's been some estimates on how much nitrogen loss occurs in these big AMZ features. The numbers are going back and forth to some extent, but somewhere between 20 and 50% of total oceanic nitrogen removal in the form of N2 gas back up into the atmosphere occurs in these oxygen-removing zones. It's not only these nitrogen removal processes that are important, there's other processes that are not directly coupled to the lack of oxygen. Nitrification, we're starting to learn more about, is an important player on the peripheries of OMZs, both above and below. And we're going to get into some of these here. So why do we care? Why do we care about nitrogen loss processes, for example, in OMZs? Because these features are changing, right? They're changing in response, based on the data, to global climate change. You heat up water, you can hold less oxygen, right? It's one of the reasons that we care about overall OMZ expansion. Another reason is that these features, especially these seasonal features, these sporadic OMZ features, sporadic oxygen loss features, are increasing in frequency throughout the world's oceans. And these are features that are driven by anthropogenic forces. The input of nutrients from the Mississippi and Choctawhattuck rivers into the Gulf of Mexico, for example. It's that input of nutrients that fuels primary production. That primary production is consumed by the microbes pulling out oxygen from the system. And there's good evidence that the scale, spatial size of these features and their frequency has been increasing in the last 50 years. And so we care about this because changing the overall global extent of reduced oxygen waters changes how we think about, for example, nitrogen loss. Okay. So people have been thinking about this for a while. We're approaching OMZ research from the context of this question about communities. Composites. thousands of microbial species interacting together in complex ways. This is what all this meta-omics is about, right? It looks at microbes at the community level. We'll get to exactly how that's done in a second. So with our work, we're focused on using these community genomic approaches to look at the structure and function of microbiology and OMCs, right? So we're going to talk briefly about meta-omic approaches, then we're going to get to the interesting stuff, the taxonomic and functional partitioning of microbes into redox gradients, and what I think is very interesting as well, this idea that there are some fairly profound differences in microbial function at the microscale, which is perhaps not surprising, but hasn't been exhaustively studied. Okay, so like I said, we focus on these two OMCs, one in the eastern tropical North Pacific, off the coast of northern Mexico, one in the eastern tropical South Pacific, off of Chile and Peru. These are water column features that we emphasized previously. Again, oxygen high at the surface decreases to anoxia. At some mid-water depth, this region here where you lose oxygen is called the oxycline, and oxygen stays low for large chunks of water column. If you kept this graph going down, it would start coming up again. But for 500 meters or so, this is effectively an anoxic body of water. These AMZ features are unique to some extent because you see nitrite accumulation in these zones. We'll talk about why later. You also see nitrate accumulation. So we know a lot about how nutrients are distributed in these zones. We know less about the microbial drivers of those distributions. So some of our work again. Okay, so sampling, we spend a lot of time on boats, as Jen mentioned. And I have this slide here to emphasize the need to think about how we sample anoxic water bodies. It's not so trivial. We do some traditional oceanographic work. We drop bottles down and collect water mass in the bottles. That's okay. It does a very poor job of capturing in situ oxygen conditions. These water bottles have to pass up through an oxygenated water column. Oxygen diffuses into them very quickly. And even if they didn't pass up through that oxygenated water column, oxygen in the plastic itself diffuses very quickly into our sample water. And so we have to employ other techniques. This is a colleague of ours filling a microcosm that we work with, bioreactors, basically just a cylinder, a glass aquarium where we can manipulate microbes. And he's filling it using water that's pumped at high speeds directly from the anoxic portion of the OMZ using oxygen. So I mention this again to emphasize the fact that if we care about thinking about oxygen effects on microbes, we have to be very critical about how we sample to avoid oxygen contamination. So we have some other mechanisms for doing this. Our lab has the collaborator has built this in situ sampler that we use for collecting microbes onto filters in the water column itself and then preserving them in situ. This is important because, as you'll see in a second, we do a lot of work with RNA. Transcripts that tell us something about how genes are being expressed. RNA degrades very quickly. For us to think about what's actually going on in situ in terms of microbial processes, gene transcription, we have to think about how these zones are actually sampled. And so we have to play around with some instrumentation to do our work. Okay, so it's not so trivial just sampling this water. And it's fun too, right? You're on boats, you fish, Josh fishes. So it's interesting. So meta-omics, we'll go briefly through this because I think you've been exposed to this before, but to some extent it's a good reminder. It's like we're thinking about microbes at the community level. So what are meta-omics? What do? We do a lot of sampling of both the community RNA and community DNA. And again, this meta simply means community. It's us taking a water sample, filtering that onto some pore-sized filter, collecting all the thousands of species of microbes that are out there in your complex water sample, and then doing a community DNA or an RNA extraction. So all the DNA and RNA from the cells pools together into one mixture. You chop that up, you fragment it, so you lose the connection between an individual gene and the genome that it came from. And then you sequence it. Right? So you end up with tens of millions of sequences potentially, maybe even more than that. Short fragments of genes, short fragments of transcripts that we then try to make sense of. Right? So this is a very complicated figure, paying attention to the details, but I'm showing this to highlight some of the steps that are involved in this process. And also to highlight one other thing. This process, the meta-omic pipeline, how we do our work is changing very quickly. This is a review paper that published last year. And some of these steps we just don't really even do anymore. Some of them completely obliterated from the way we process our samples. But some of them are remaining important and becoming even more important. So I emphasize a couple of things here. The sample collection that I've already talked about, the idea of making sure that we can accurately capture in situ processes, very important. And then this final step, making sense of those received fragments, the informatics. That's the part that's the hardest. Making sense of the data in some meaningful way, extracting the full amount of information content of it from it. Very challenging. We have a lot of ways that we can potentially do that. This is a brief schematic of our computational pipeline. Again, we start with millions of sequence fragments. We filter them for some quality threshold. Then you can process your data in a lot of different ways. You can search for known genes that your sequences are related to out there in public databases. This can be informative to some extent. Or you can do some assembly-based approaches where you try to map up your fragmented pieces of RNA and DNA into longer contigs. That can be really useful too. It can link genes together in a chromosome. Again, that information was lost when you chopped up your sample. And then you can do some comparative genomics. You can compare your sequences to bigger contigs that are out there in the database. You can also do some stuff that doesn't require comparing your sequences to something in your database. This is the harder work, but it's still possible. You can cluster your sequences based on sequence relatedness and look for patterns in the clusters of related pools of sequences. But once you do all of these steps, there's a lot you can do in terms of pulling out information. You can assign function to your gene fragments or to the full genes in your assembled content. You can assign at least an estimate of the taxonomic group that those genes came from, which can be very important. You can map them together, start building metabolic networks of your genes. And you can do other things as well. You can calculate bulk characteristics of the community that you sample. And so we've done that just as a starting point. This is data looking at metagenome sequence fragments. As you go down in depth in the Eastern Tropical South Pacific, the blue is the OMZ region, the region of low oxygen. What I'm emphasizing here is a decline, a consistent decline that you see with oxygen in terms of the diversity of the microbial community that you're looking at. So if you're looking at how many different taxonomic types there are in your sample, ask the question, how does oxygen affect the complexity of that community? There's some very consistent patterns. Community complexity decreases as you go into the center of the OMZ. We're not the only ones that have seen this. This is a paper by Michael Beeman's group looking at the Eastern Tropical North Pacific, doing essentially the same thing, counting up the richness, the total number of taxonomic units that they observed over oxygen gradients. And they saw something similar but slightly different, this unimodal distribution where above a certain oxygen threshold, both below and above the OMZ, community richness tends to decrease with oxygen content. Whereas if you look at the region of low oxygen concentrations, community richness decreases as you remove oxygen from the system. So quite different patterns, and this is interesting. It suggests something about the complexity of functional processes in the OMZ. Right? And so we've looked into this as well. What's the distribution of different niches? Functional richness as you go from regions of higher oxygen to regions of lower oxygen. There's a lot of ways to think about this and some caveats to how you should think about functional richness. We did it by simply counting up the number of different protein types that are out there in metabolic categories. And it's consistent with what you would expect from the taxonomic distribution as you go from the oxycline environment where you see a peak in functional richness into this harsh OMZ environment where the environment becomes more uniform. The number of niches, at least expressed through this metric, decreases. Which I find interesting. So that's kind of the community level process that you can try to, or structure that you can try to infer from, in this case, metagenomic data. You can also look more closely at individual taxa. This is data from the Eastern Tropical South Pacific again, 4 different depths, 50 meters, and then three depths within the OMZ itself. And this is just highlighting some of those taxa that are most transcriptionally active. So we've assigned taxonomic identities to the gene transcripts for our data sets and have looked to see which groups are actually out there at community level, exerting the most effect, right? So these tails, which is showing the top 15 taxa or 10 taxa, these tails go out into the thousands, right? These are complex communities, thousands of species interacting together. But just thinking about who the most dominant players are can tell you something about key functional processes, right? So I think it's interesting that up to 20% the entire transcripts that you recover, for example, this death in a community of thousands of individuals, map to this one specific organism. In this case, it's an aerobic ammonia oxidizer. Here's that first step in the nitrification process. And it's this archaeal member, this nitrosochemose. Very interesting. You see it in the surface waters, at the oxyplant, and even into the low oxygen waters. So nitrification, based on transcript abundance, is very important at the surface, or at the OMC groups. Other groups take over, though, as you start going down into the OMC. Some of the notable ones I've highlighted in color here. The one in black here is quite important. These are genes or transcripts that are mapping to this organism. This organism called Coenia. Coenia is one of several different groups of Planktomyces bacteria that carry out this process, the anox process. Again, the anaerobic oxidation of ammonia that removes N2 from the system. So it's nice to see this. We knew that anox was important based on process rate measurements. We didn't have a good sense of just how important anox bugs are in activity compared to everything else that's out there. So this type of data can tell us something about that. You can also see some other interesting groups that come up into abundance, including these purple highlighted groups. These are bugs that oxidize hydrogen sulfide as an energy source using nitrate as a terminal electron acceptor. Stripping the energy out of that process and using it to fix carbon. Right? So these are bugs I'll talk about in a second. Not so rare at the surface, but seem to have an important role at the center of the OMC. So that's data, transcript data, that's focusing on the level of taxa. You can also use that type of data to look at individual genes. So these are just different marker genes at 3 different depths into the OMC for key processes that we care about: ammonia oxidation, nitrate oxidation at the surface, nitrate reduction process, and then the anox process. And they're color-coded based on the taxonomic identity of those genes. So we can not only get a sense of the distribution of those genes, but we can look into their taxonomic profile, get a sense of who are the dominant players that are contributing to these processes. Genes like nitrate reductase, that's a community level process. A lot of bugs can produce nitrate as a terminal electron acceptor. Every major group can do this to some extent. If you look at the process of anox, for example, that process is dominated by a very specific taxonomic player. In this case, just those bugs that are related to known anox bacteria, Coenia and organisms. So this type of data can tell you something about the complexity of the gene signals that you're getting. It can also tell you something about the importance of thinking about different data types. So this is a graph that's showing DNA data and RNA data. Also, again, the same site going down in depth, looking at the relative abundance on the x-axis for different genes, 2 genes. Of nitrogen transporter gene, which is involved with getting ammonia into cells. And I show the distribution and the abundance of those genes for both the DNA pool and the RNA pool. And I show this graph a lot because I think it is a functional input taxon of nucleides. So if you look at these pie charts, they're color-coded based on the domain of life that is contributing to those samples, either bacteria or archaea or classified samples. You might think that your community, for these 2 proteins, is fairly diverse. Well, because there is some level of diversity, right? It's very different if you look at the RNA pool, especially for this ammonia monooxygenase gene here. It looks like it's a composite of both archaea and bacteria and wood. If you look at what's actually being expressed, it's entirely archaeal signal. So I think this is very important. It can tell you something about the relative activities of different players in that system, in this case 2 players in key steps of the nitrogen cycle. You can also use this type of data to pull out linkages that you might not have thought of as being present based on biogeochemical evidence, for example, but there's still an important contribution to the signal, but perhaps a big group. So this is data highlighting the distribution relative abundance, sorry, on the y-axis, of gene fragments that map to known sulfur-oxidizing bacteria and known sulfate-reducing bacteria in OMZ systems here and non-OMZ systems here. And there's a strikingly higher and obviously a significantly higher abundance of these sulfur-oxidizing bacteria and sulfate reducers in OMZ systems. That's perhaps not surprising, but it's a little bit surprising for these systems because sulfide doesn't accumulate here. If we hadn't had genetic data to go in and see this signal, we might not have suspected that these functional types of bacteria play a key role in the system. And it begs the question, well, if there's this genomic evidence of bacteria, especially sulfur oxidizers, many of which are related to this clade SUP05, which has been shown to be related to chemosynthetic sulfur oxidizing symbionts from deep sea plants. It begs the question, how are these organisms participating in elemental cycling in the system? Where are they getting the sulfide that drives this process? So we've done a little bit of work with our collaborator Don Canfield to look at this process, thinking about the potential for sulfide generation by active sulfate reduction in the water column, something that hasn't been shown before in these sites where sulfide doesn't accumulate. Turns out if you do radiolabel experiments with radiolabel sulfate and look for the production of sulfide, you can actually see it. You can measure sulfate reduction of labeled sulfide in these OMZ settings. But you never see sulfide accumulate because it's immediately consumed by that robust community of sulfur oxidizers in the water column, with nitrate as a terminal electron acceptor. Again, a direct coupling between the sulfur cycle and the nitrogen cycle. So the point is that some of that meta-omic data can shed some light on potentially cryptic elemental processes that you can then draw hypotheses about and go out there and test with great data. Okay. So you can also, again, look at these coupled RNA DNA data types to get a sense of who's most active where. So this is data that's breaking up RNA and DNA reads based on the taxonomic affiliation with the DNA data on the x-axis, RNA data on the y-axis, and the one-to-one line here. And where you are on the relative side of that one-to-one line tells you something about how active you are as a taxonomic unit, right? So there's a lot of bacteria and archaeotypes that have relatively low abundance in the DNA fraction, not a major numerical component of the community, but their RNA expression is very high. They can, in fact, sometimes dominate the entire transcript pool, which I think is very interesting. It tells you something about the potential for low abundance, low frequency functional types to still be doing something meaningful in a functional perspective. Okay. So you can get some sense of the relative activity over vertical gradients, that type of data. You can also get some sense of variation within the taxa, depending on where you are in the water column by using transcripts data. And so this is data that's showing transcripts that map only to this SUP05-like clade. Again, that's the sulfur oxidizer that I talked about a second ago. And it's showing all of, well, at least a large subset of the genome, the genes in that genome and their transcription level, breaking it up by those that are enriched in the OMZ core and those that are enriched in the upper OMZ. And of course, there are differences, right? That's not surprising. Bugs hang out in different places. They turn on different genes at different times, depending on vertical gradients. But we can use metatranscriptome data to try to get a sense of which genes are turned on where. The point of this graph, again, details are super important, is that all the genes involved in sulfur oxidation turned on essentially only at the OMZ core. You could still find these SUP05-like organisms at relatively high abundance in the upper OMZ, but not really expressing a lot of sulfur oxidation genes. This graph doesn't necessarily highlight that, but it's true. So you can get some sense from transcript data about vertical gradients in microbial niche. Right? So we're doing that. We're also doing some other things that are trying to get a better estimate of exactly what are the environmental conditions that would limit some of these key processes everywhere. In this case, I'm going to talk about data related to nitrogen cycling. In this case, we care about what is the effect of very subtle changes in oxygen on the, for example, production of N2 gas. And so to get at some of those questions using meta-omic data, you have to couple it to process rates. Very important. It's perhaps a truism, but it's challenging to do logistically. And so we have to do things like work in microcosms. So this is work that we did with our collaborators at Dallasgard in Denmark, using these essentially big glass cylinders that are equipped with highly sensitive oxygen sensors, are equipped with inlet and outlet ports that we can manipulate the oxygen conditions with. And we can do it in such a way in using sensors that have high enough sensitivity to actually look at oxygen fluctuations at the level that these bugs are likely to experience in situ. Which is again at the level of the nanomole, which for most oxygen sensors is very hard to work with. So this is the design of some of these experiments. We take water from the OMZ, we pull down the oxygen or any that was introduced during sampling, and we can manipulate the oxygen throughout the time course of this experiment. That's over a day or day and a half in length. We can keep oxygen very low, as shown in this graph here, effectively below 20 nanomolars throughout the course of the experiment. Or we can manipulate it in other ways. We can make it high at the beginning, drop it down, or vice versa. And at the same time, we can couple that oxygen manipulation to measurements of process rates. In this case, the production of N2O gas by nitrification, or the production of N2 by anoxic, or the production of nitrogen gas by nitrification. We can use this data to get some sense of what are the actual thresholds that turn on or off these processes. Right? For oxygen concentrations that we actually care about in the environmental setting. And so we've done that with, again, collaboration. Mostly Kay has done the bulk of these calculations, looking at what are the inhibition thresholds for denitrification and anoxic in particular. Right? So if we look at our data, denitrification, production of N2 and N2O gas, is 50% inhibited at what is a fairly low oxygen concentration, 200 to 300 nanomolar. The anoxic process, the less sensitive, 50% inhibition at effectively 1 micromolar. I highlight these because this is very different than what previous studies have shown. Previous studies, perhaps not with the same level of resolution, have put some of these thresholds at up to an order of magnitude higher. And that matters, right? If your threshold is a lot lower, thinking about the production of N2 gas through these processes, that changes overall magnitude of perhaps what you would want in terms of N2 loss, right? Thinking about total volume of water that are exposed to the oxygen conditions. Anyway, so thinking about oxygen concentrations in the context in which they are actually relevant in situ is critically important. And so that stuff is doable, but what does it look like when you couple those process rate characterizations, those measurements, to what we do? The analysis of gene transcripts, for example. And so we've done this for those microcosm experiments. What I'm showing here are different genes with key steps of the denitrification process, and also key steps of nitrification, and key steps of the anoxic process here at the endpoints of these experiments. And do they correlate with oxygen conditions? And these things are colored or shaded differently, essentially according to low oxygen treatments, and then lighter colors reflect increasing levels of oxygen. So the question here is, what can we infer anything about process rate characterizations from metatranscriptome data, for example? It's a tough one. Sometimes you can, sometimes the signal is very clear, a lot of times it is absolutely not clear. Highlighted a few cases where it is clear. These are genes, these last two genes here, genes that convert nitric oxide to nitrous oxide and nitrous oxide to gas, two terminal steps in denitrification. Here, there actually is a pattern. You would expect their expression to go down as oxygen increases. That's true. You can see that in our data. We can couple these community-level transcript approaches to these process rate measurements in a way that actually makes sense, at least for some of these genes. A lot of others, the pattern doesn't make sense at all. Right? So I highlight this in part to say that there's potential in trying to make these linkages, process rates, meta-omic data, in an experimental setting, but there's a lot of caveats as well, especially when you're thinking about trying to extrapolate signals from thousands of different genes. But there's some potential there. Okay. So this longer part of the talk highlights some conclusions that we've been some newer data. Steep OMZ gradients in oxygen and substrate availability, including at that nanomolar scale, over sometimes most effectively the meter scale. Great vertically structured microbial index. That's perhaps not surprising. But what other factors drive OMZ microbial structure and function? And our OMZ functions, some of the ones we care about, partition differently amongst microhabitats, specifically between those communities of microorganisms that are free living out there in the water and those that are attached to surfaces. So this is something I'm very interested in, and it relates to something that as a community of real oceanographers, we don't do so well these days. Perhaps we've never done it well, but we've been thinking about it for a long time. But what we don't do well is sample the complexity of the ocean water column in the way that it actually exists. We tend to collect bulk water samples and treat them in a uniform way, making the assumption that microorganisms are distributed more or less evenly throughout that water column, and that your liter of water contains no spatial heterogeneity or no chemical heterogeneity. That's absolutely not true. This is a cartoon from a paper by Roman Stalker a couple years ago in Science, thinking about what microbes actually see out there in the water column. And it's a completely heterogeneous environment. There's particles all over the place. Live zooplankton, sinking phytoplankton, degrading phytoplankton in various states that are emanating plumes of dissolved organic carbon that are diffusing away at different rates. There's a bunch of microorganisms that are distantly, spatially away from these zones. A bunch of others that are only localized to the plume zones away from sinking particles. There's others that use chemotaxis actually, and then adherence functions to latch onto the particles and degrade them. This is a very spatially complex environment, not only considering particles versus non-particles, but also the distribution of micro-niches within particles. A lot of chemical heterogeneity, especially oxygen heterogeneity. Relatively high at the surface, but low within the particle, perhaps. So we're interested in this question. Can we apply meta-omics, community-level approaches to think about microbial structure and function, to this question of the micro-niche? Are there differences in who's there, and more importantly, what they're doing? And people have been thinking about stuff on particles for a long time, not necessarily at the level of community functional approaches, so this is where our approach takes a slightly different direction. So this is data from my graduate student, Sangeeta Ganesh. She's worked quite a bit on this and is continuing to pursue her thesis on this topic. In this case, we're looking at differential distribution of key taxonomic groups, bacterial groups, on those that are enriched in the particular fraction and those that are enriched in the free living fraction. And there's significant differences, and differences among taxa that we might expect to be enriched in a particular fraction. For example, planktomyces, they like to adhere to surfaces, they're swarms to some extent. And also things like delta proteobacteria. A lot of deltas are anaerobic, a lot of them are sulfate reducers, and lo and behold, you find them enriched on particle surfaces. So some of these patterns make a lot of sense in terms of the taxonomic distribution. What I think is even more interesting is just how important that separation is, particle associated versus non-particle associated, especially compared to the steep vertical gradients that we see in OMZ water columns. And this is some of the data to highlight this. This is just a PCA plot that's relating different samples based on the community taxonomic profile. Circle the ones that are samples reflecting the particle-associated fraction, and those reflecting the free living fraction in different colors and different depths. What I find striking is that even though you sample across a gradient that goes from 200 micromolar oxygen in the water, surrounding water, to one where it's effectively anoxic in the core of the OMZ, and also you encompass steep gradients of nitrite and nitrate and other key drivers of environmental microbial function. Perhaps the strongest driver of what your microbial community looks like is actually whether or not you're attached to a particle. So it's a key differentiator of microbial ecotypes, perhaps even stronger than some of the vertical gradients that we sample. And the microbes are distributed differentially between, or specific microbes are distributed differentially between different fractions as well. And this can be interesting. It can tell us something about different ecotypes within one type of microbial genus. So these are just three different microbial taxa that I've highlighted here in the depth gradient, their relative abundances, in both the free living and the particle-associated fraction. Their abundances differ not only within the fraction, but also the differential abundances as you go down in depth, suggesting at least the possibility that there are different, like I said, microbial niches, subtypes within microbial groups that are perhaps adapted to being attached to a particle, not attached to a particle, or at least at some level. Okay, so what does it actually look like if you ask the question, what are the key functions that are enriched on particles versus those that are enriched in the free living environment? We look at a lot of different functions in our data. I've just highlighted a few of the most enriched in each fraction. These are the ones that are higher on particles, these are the ones that are higher in free living. And a lot of it makes a lot of sense. So what I've highlighted here are genes that are involved with microbial social interactions to some extent. These secretion systems, often used with getting products from one microbe into the cell of another microbe, for example, or secreting a virulence factor or something along those lines. They're also often used in motility. Motility is also enriched in general in the particles. Adhesion is enriched. Signaling, communicating with your neighbor, is enriched on particles. Resistance to antibiotics and toxic compounds. Those functions are also enriched on particles. Things that are enriched in the free living realm tend to be things like transporters. Right? Devoting a lot of energy and resources to getting things into your cells from a potentially dispersed environment out there surrounding you. So I think this is really interesting. It's perhaps not surprising. We're effectively comparing microbes living in a condo development, a biofilm to some extent, very close in proximity to their neighbors, versus those that they only come in contact with their neighbors in free. So some of these functions make a lot of sense. What about some of the key functions that we care about in terms of OMT processes? So these are data, again, some of the same nitrogen-related genes that we plotted before, different columns or different depths going into the OMT, left to right. And they're color-coded or shaded based on their relative abundance in the particle-associated fraction and the free living fraction. Some of the patterns are all over the place. You can't really say anything. Some of them are very consistent, though, and I find that interesting. Especially these two, these genes that are enriched in the particle fraction. These are, again, these genes, nitric and nitrous oxide reductase. These are two genes that carry out those two terminal steps into nitrification. The gas-producing steps into nitrification. Not so abundant in the free living fraction, the gray fraction. All in the particles. Another thing this highlights is the taxonomic identity of these genes. That's what all the different colors are. For denitrification, we know that the microbes that carry out these processes it's not all done by the same microbe. It's a community of interacting species that carry out these different steps. Nitrate to N2 gas. Okay. I think this highlights the potential that there can be microspatial differences in where these organisms are, and potentially, therefore, the extent to which, the strength to which they're coupled. Okay, so that's just gene distribution, right? Omics. More importantly, or equally importantly, can we assign process rate measurements to that microspatial distribution? Sangeeta is working on this question with our collaborator, Laura Bristow, who's a postdoc in both Amdrik's lab in Denmark, asking this question: what is the microspatial variation in process rates that we care about? So this is data that basically just highlighting oxygen and nitrate conditions at the site that we're doing this. This is off the coast of northern Mexico. And there are some interesting patterns. Some of them are consistent with the Meta-omic data. Some are not. So here we're just highlighting, again, key nitrogen processes. In this case, ammonia oxidation and nitrate oxidation. These processes, at least if you look at process rate distributions, they compare effectively just the blue data, which are those processes or those rates that are restricted to the free-living fraction, the small fraction, versus those that are on particles, which are the green. These are evenly distributed, or at least within the context of our sample, relatively evenly distributed. There's others that are predominantly particle-associated. Nitrate reduction, for example. Nitrification, some steps. Analogs looks to be evenly distributed, depending on how you analyze it. So we'll get to that. So let's take a slightly more in-depth look. So this is nitrate reduction, looking at rates versus gene transcripts. The number of different gene or transcripts encoding nitrate reductase that we find in our gene pool. How do these two compare? You look at the rates. This process seems to be confined exclusively to the particulate fraction, at least within the OMC depths that we sampled at. Very different than if you look at the transcript abundance. It seems to be suggesting that at least the expression of these genes is relatively even between these fractions. That's an interesting pattern. I don't know quite what to make about this discrepancy, but it highlights some key differences. There's a decoupling between nucleic acids and their relative opponents and the process rates that you may care about. Denitrification, at least 2 steps of denitrification that I mentioned before, the 2 gas-producing steps. These are more consistent, which I'm happy about. So NOR-B and NOR-Z genes, these are confined again primarily to, or the transcription of these genes confined primarily to the particulate fraction. And lo and behold, the rates of these proteins that you're able to get rates on, those seem to be confined primarily to the particulate fraction as well. So at least for a subset of genes, rates and genes correlate with one another. It's reassuring. There's the part. Analogs, not so much. Again, rates suggest that this process is fairly evenly distributed between fractions within the O and Z guts. But there's various depending on how you look at it, whether or not you add 15N ammonia to measure analogs production of N2, or whether or not you start with 15N nitrate. It makes a difference. I'm not sure why it makes a difference, but it does, and we're thinking about this. But then, both of these are very different than what you see if you look at the gene fraction. This is a marker gene analogs that's differentially much higher in the free-living fraction. And this is something that we've seen repeatedly, not only in the Eastern Tropical North Pacific, where we've done this recent work, but also in the Eastern Tropical South Pacific. It's also something that other groups have shown as well. Not sure what to make of this discrepancy between gene distribution, in this case transcript distribution and rates. But again, it highlights this important component. The meta-omic data is not the entire story. Neither is your process rate. Okay, so those are some key functional genes that we care about in the context of O and Z processes. I'm going to end by just highlighting a couple of other genes that I find interesting that we've also seen in this particular story. And these are DNA mobilization genes. What I'm showing here is different depths going down into the O and Z. This 1,000 meters is below the O and Z, so it's oxygenated again. And this is the distribution of these genes called transposases, or genes encoding transposases, and genes encoding integrases, both in the free-living fraction and the particle-associated fraction. And there's a big difference. It's not only in these 2 types of genes that I point out, but a lot of other DNA mobilization genes as well. Genes that are involved with snipping pieces out of a genome, moving a DNA fragment to another spot in that genome, or to another spot in some other cell's genome, are enriched in the particle-associated fraction within the biofilms, which I find very interesting. And we can think about some hypotheses for why that might be. Maybe it's a very simple answer. Maybe it simply has to do with surface attachment, facilitating spatial proximity, so you're closer to your neighbors, easier for you to exchange genes. Gene transfer is more rampant, close to your neighbor. To some extent that makes sense. We can see this pattern in processes as well. There could be other things going on as well. I mentioned before that these particular environments are very heterogeneous on particles. Not only in gradients of oxygen, for example, if you're outside the O&Z, but also gradients of other redox important chemicals, and also gradients in taxonomic composition, right? So compared to the water column, the particular environment is very heterogeneous. Spatially, but also temporally, especially if some of those particles get consumed by a zooplankton, for example, pass through gut, exuded out. There's a lot of temporal heterogeneity in which microbes attached to those particles may see. And so some of that heterogeneity can actually drive for, or impose some selection pressure for the ability to diversify your genome. That could be something this way. Or it could be simply that particle-associated microbes have a smaller, lower effective population size. We don't need to get into the details of this, but that can affect the ability to create harmful elements. Anyway, this pattern is very interesting, and to some extent it's a large-scale pattern. We show this for O&Z data, but you can also go out there to Hawaii and sample over not 0 to 200 meters, but 0 to 4,000 meters, and see a very similar pattern in terms of the distribution of transposases. So what I think this is saying is actually quite interesting is that the dynamics of gene flow, potentially, population structure, and also genome content, change not only with depth, but also at the microscale, depending on how close you are potentially to your neighbors, and some of the selection processes operating on that unique microbial micro niche that you're in. So we're doing some stuff to follow. Okay, So some broad conclusions from this. Vertical environmental gradients create high levels of community metabolic complexity, structure, taxonomic and functional regions, declining at suboxic depths. Remember, if you remove oxygen from the system, the community gets less complex. OZ microbes play diverse roles in elemental cycles. Highlight a few of those that we care about. There's a lot of others that remain cryptic, so we're working on some of those in our lab. We're starting to as well. A couple of meta-omic and process rate measurements under in situ conditions can highlight nanomolar sensitivity of some key enzymes and key gene expression processes. Right? We care about these levels of oxygen because that's what these bugs actually see out there. And surface attachment, despite these strong vertical gradients, is an equally strong, or perhaps even stronger determinant of diversity of function compared to vertical. Key OMZ functions are partitioned between microhabitats. I think it's very interesting. It implies a coupling of some of these key processes that we care about. Particle-associated communities finally may be hotbeds for genome rearrangements, maybe via transcription. I think that's really cool. It says something about the spatial environment of microbial genome reshuffling out there in nature. That has direct implications for how we think about the distributions of key biogeochemical properties. Okay, so shifting ocean conditions finally will likely affect the distribution of biogeochemical importance of microbial. Right? All these are dynamic, especially at their surfaces. The distribution of oxygen will affect where the microbes are, and also, as we've shown, when they start doing it. Okay, so I'd like to thank a few people, everyone that actually does the work, the lab members, Corey, Josh and Sangeeta have been on several cruises. My postdoc Neha, excellent postdoc in many different ways, and a lot of this work. She's not working on these OMZ systems, she's working on the Gulf of Mexico OMZ system, but and my collaborators, some of them internationally, Bo is particularly very similar work with and it's excellent. Some collaborators. And finally, funding from NSF, the Sloan Foundation, and the Agron Institute and the War Foundation for their those differences. Thank you.
All right, so maybe you can do questions.
Is there any evidence that these communities with levels of mobilizing elements, that they're actually mobilizing changing or starting with?
Yeah, no, we are starting to think about how we would do that. And the best way to do that I can think is this small sample of series the same ecotypes, same strain types, part of the fraction, and actually trying to capture some of those effects. We don't have the sampling network in place to do that quite yet. That's what I want to do. So right now, we're basically for the expression of these genes. I'm trying to relate some of those back to overall genome characteristics of where they're coming from, genome size, for example, and get some preliminary hints on that, but it really will take some time. Yeah.
Great presentation. Your results illustrate the lack of agreement among DNA and RNA and probably proteomics and rates. You must have given that a lot more thought. What do you think's going on and which is correct? If we know which one of those is correct, then we can calibrate all the others. Which one is correct? Or what's going on?
I'm sure there's a lot going on. But some of what's going on is a disconnect between common sample to some extent. You have a disconnect, at least between the macromolecule rules simply because of rates of turnover, right? Does that make sense? Proteins stick around for a long time, transcript very quickly. And also there's a lot of inter-species differences in how much transcript of protein you need to carry out a function. And interspecies is not the right word, but intermetabolome differences. Some processes are very efficient, others are not very efficient. So expect to see a correlation between, say, protein abundance and transcript abundance for certain types of processes that are turned over at certain rates and others that are not. And so that kind of, within genome complexity, in terms of enzyme function and efficiency, is just imposes a lot of stuff onto the system that I don't think we have great handle on yet. In terms of what's correct, it depends on what your question is, of course, right? It really does. I don't think I can advocate for just working on process rates, right? Because one of the big caveats that we have here is that we take a system, a body of water, bring it aboard ship, and manipulate it in some way to get that process rate characterization. That has its own level of bias, and it's a big level of bias. We've done some work on the scale of bottle effects at the community level. We do that with OMP water, and it's big. And so, yes, process rates may be important if you think you only got function, but in situ process rates is the next step, so maybe that's the next step.
You avoid the controversy, you just measure one thing. You don't have to worry about complaining.
And you get 0 rates for cryptics.
And it also depends on the time scale at which you care about this, right? And so if you're interested in the kind of time integrated processes, maybe just looking at the meta genome is appropriate. I don't know.
Great. All right. Oh, one more question, and then we better go upstairs.
So, might be my basic question, but how quickly do microbes trade genes? And if they do it really quickly, how can you determine who those genes belong to? If you're trying to say like a particular strain or a gene.
So the question of how quickly? Very quickly. It's not hard to find good examples of horizontal gene acquisition that happen just as soon as you start mixing microbes. It can happen very quickly, and you track who it comes from by genetic similarity. So hopefully you have a system that you can acquire enough genomic data for to actually capture the horizontal gene transfer event. And hopefully that horizontal gene transfer event will have enough of a signal of donor-like elements to be able to extrapolation.